Module 9, the peripheral nervous system. The sense of smell. It's time to study the special senses. The sense of smell is also called olfaction. In this mid-sagittal section of the nasal cavity, you can see the olfactory epithelium in the olfactory recess. Here is where the receptors for the sense of smell are located. Can you see how they are located in the upper part of the nasal cavity? That means that you can only identify smells in that spot, not all over your nasal cavity. In this close-up diagram of the olfactory epithelium, you can see olfactory bulbs that are immediately superior to the cribriform plates. Cribriform plates are bones that provide protection for the olfactory bulbs. They have small holes called olfactory foramina, which let the axons of the olfactory neurons travel through so they can reach the olfactory bulb and synapse with association neurons. The other ends of the olfactory neurons are exposed to the air in the nasal cavity, and that's where the olfactory vesicles are that end in olfactory hairs. The olfactory hairs are surrounded by a mucus layer. Looking at the neurons, you can see that they are bipolar neurons, having one axon and one dendrite. Now, all of these parts, the olfactory epithelium and the neurons, are continuously replaced every 2 months or so. When a cell is lost, a basal cell replaces it. This is a special situation. I've already told you that neurons cannot be replaced. Well, olfactory neurons are an exception to this rule. So, how are you able to smell? First, chemicals have to be airborne. If an item does not send chemicals into the air, then it will not create a smell. Do you know that new car smell that people love? Well, that is due to the chemicals in the new fabric, plastics, and other materials of a new car that deliver molecules that are airborne. After a few months, those car parts don't release chemicals into the air, and that new car smell goes away. Well, a substance that evaporates into vapor to become airborne is called a volatile substance. So those are the only types of substances that we can smell. But there are some volatile substances that we cannot smell. That's because in order to identify a smell, an airborne chemical has to make it to our olfactory hairs, and that can't occur unless those chemicals can get through the watery mucus layer. That means that a substance has to be at least somewhat water-soluble. Additionally, a substance has to be somewhat lipid-soluble in order to move through the plasma membrane. After a substance makes it to the olfactory hairs, receptor molecules in the hairs will bind to the substance. Then a chemical reaction occurs. If there's enough of that reaction, then action potential is generated. So if a smell is too faint, you cannot smell it because there are not enough airborne chemicals around to generate an action potential. If an action potential is generated, the signal travels along the axon to the olfactory bulb, where there is a synapse with a specialized association neuron that modifies the information and then transfers it to the brain. Interestingly, scientists don't fully understand this process. Once, they thought there were 7 different types of receptors that, based on the combination of their responses to airborne chemicals, would give us the ability to identify all the smells we can smell. But this idea is changing because of new information showing that there may be up to 50 or more primary sensations. That means that there would be 50 individual receptors that can bind to an airborne chemical. The smell we identify then is a result of the combination of the responses of those 50 receptors. Regardless of the number of receptors we have, we do know that the threshold for them is low. It doesn't take a lot of stimulation to trigger an action potential on the axons. That means that the concentration of airborne chemicals in the air does not have to be very great. Thus, we can identify fairly faint smells. But you know that our olfactory receptors are able to adapt pretty quickly. That means once the olfactory hairs have been stimulated for a bit, they won't continue to send action potentials and you stop smelling the aroma.